Greetings everyone, and today we are going to discuss about the third line of defense. Third line of defense, first and foremost, giving the intro. What is third line of defense? As we have previously studied, what is the second line of defense? Second line of defense says to us that if uh, you know pathogen is crossing the first line of defense, then second line of defense will be acting as a non-specific line of defense to attack uh, the pathogens by phagocytes as well as by cytokines. So as a result of this, this occurs in second line of defense. Third line of defense is a specific line of defense, which is also known as adaptive immune system, adaptive immune response, as well as why is it known as adaptive or acquired immune system? Because it is acquired after birth. It is not by birth. It is not by inborn immunity. It's by birth. It's acquired, so it is not adaptive immunity. And here it is dependent on. It acts on the influence of second line of defense. And apart from that, it is specific because it works on the basis of a specific antigen containing pathogen which has entered the cell. Now, coming to before discussing uh, what does it actually include, it basically includes two major parts. First includes a cell-mediated immune response, cell-mediated immune response, as well as antibody mediated immune response, which is also known as humoral response. Basically, if uh, as we know that antibodies are released within the blood stream, so these antibodies, when transferred from one organism to another organism, for suppose one organism is immunized, other is non-immunized, so when immunized and person's antibodies are transferred from its bloodstream to non-immunized person artificially, it will cause the non-immunized person to become immunized as well. At that moment, this will this you know trans this immunization process occurred due to the transfer of humor word refers to the body fluid, as a result of which at that time when scientists came to know about this type of immune response, it was then named by those scientists as immune response. But we can also call this as antibody immune response, antibody mediated immune response. And here, uh, let us, before discussing about cell-mediated immune response and humoral immune response, it's important to discuss what is the structure of MHC. We know that over within our body, we have the largest gene locus, which is MHC. MHC is present in all nucleated cells. There are two classes of MHC. First includes the MHC class 1, MHC class 2, and these produce the and set of receptor proteins which are known as human leukocyte antigens, HLA, human leukocyte antigen. And first coming to the part of discussing MHC1, MHC class 1. Discussing with its structure, MHC class 1 is basically, first and foremost, MHC is located on the sixth number of chromosome of our human DNA and within the cell. And here, uh, MHC class 1 here consists of uh, you know it is consisted of a quaternary structure. It is having two major components, alpha chain and beta chain. Alpha chain is divided into three subunits, alpha 1, alpha 2, alpha 3, which is having disulfide bonds, as well as beta chain is beta includes a single microglobulin subunit, microglobulin subunit, which is beta 2 microglobulin subunit, as mentioned in the diagram. As you can see, alpha 1 and alpha 2 chains have a depression. This is kind of depression between alpha 1 and alpha 2 beta chain, which to which the particular antigen binds. While when it comes to the alpha 3 chain, it is alpha 3 is parallel to beta 2 microglobulin subunit, as a result of which it causes to give this structure uh, to the MSC class 1 HLA. At that moment, the structure, this complete structure of alpha 3 chain is you know alpha 3 chain is transmembranely, it is embedded in the transmembrane, so it is a transmembrane protein. So here it is transmembrane protein. It is attached within the plasma membrane, so that it forms a, it gives it firmness and attachment. On the other hand side, this is the basic structure of MSC class 1. MSC class 1 is present on all nucleated cells of the body, as well as a non-nucleated cell which is known as platelets, but it is not present on antigen presenting cells such as macrophage. T helper cells, B cells, as well as dendritic cells, dendritic cells, macrophages, T helper cells, and B cells, because they contain another type of MSC class which is known as MSC class 2. Now, coming to some points here, that whenever our body is having, for suppose there's a cell, if there's a cell which is affected by any sort of pathogen, let's suppose a virus is entered. So when virus is entered, it's in the cell, then our body will start to, then first and foremost interferon will start to release out of it, due to which it will call foreign to will begin to call the phagocytes to attack over that cell, as well as when phagocytes will attack over this cell. Let's suppose interferon is released in this condition. Let's suppose which interferon alpha, because there are three types of interferon: alpha, beta, and gamma. Alpha and beta are released by virally infected cells, and gamma interferon is released by natural killer cells, T helper cells, and cytotoxic T cells, which have another function as well we have discussed previously. So here, as interferon alpha or beta, any one of them is released, natural killer cells arise, attack this cell, and cause to represent the antigen. Now antigen is a particular particle of a protein. It can be a protein as well as a nucleic acid. It can also be a carbohydrate molecule, polysaccharide molecule, and in order to become a nucleic acid, it needs to be either a nucleoprotein, and in order to be a carbohydrate, it needs to be a glycoprotein. So a glycoprotein or protein or nucleoprotein, or a lipid molecule, if it a lipoprotein is supposed to attach, then this antigen is going to attach over the complex, the HLA of MSC class 1 of natural killer cells, which is known as, which is present over here. Let me just try to draw it as well. Now what actually will happen over here is that here this antigen will be represented over MHC class 1, and once it is represented over this MHC class 1, this MHC class antigen represented over the MHC class 1 will be taken back, will be taken and to represented over, actually there is a complete mechanism here, that here when natural killer cells are having, they are also having MSC class 1. So when they will have the antigen of that particular pathogen attack over them, they will represent that antigen over that particular over their cell surface of MSC class 1. Now when natural killer cells will be coming, natural killer cells will utilize their receptor, which is known as CD56 receptor, in order to detect that particular type of antigen. And once they detect that particular type of antigen, they will recognize that their MHC class 1 is either altered, or it is lost, or it is modified, due to which we need to create an immune response against this cell. So once it creates immune response, destroys the cell, then represents that antigen on its MHC class 1, where this MHC class 1, which is represented over the natural killer cells, uh, human leukocyte antigen, will be now represented over another cell, which is known as the helper T cell. Now of course this the next step we are going to discuss when we are supposed to discuss MSC class 2. First, MSC class 2 here includes, it is a heterodimer structure, which means it is a heterodimer structure. It includes two different dimers of uh, proteinaceous globular proteins, which includes alpha chain and beta chain. Alpha chain consists of alpha 1 alpha 2 subunits, and beta chain consists of beta 1 beta 2 subunits. One more thing, that here there is a disulfide bond which causes to attach them, and alpha 1 and beta 1 chain are parallel, alpha 2 and beta 2 chains are parallel. Alpha 2 beta 2 chains act as transmembrane proteins. They remain within, attached within the attached within the plasma membrane, such that when they are they are embedded within the plasma membrane to provide it rigidity. And alpha 1 and beta 1 chains are having depression or groove, due to which they form the binding group to which the particular antigen is presented, or inhibitory receptor is attached. So this is how the structure looks like. Um, Now MSC1. MSC1 is present on all the antigen presenting cells, such as macrophage, helper T cells, dendritic cells, and B cells. Moreover, antigen present these MHC class 1 interacts with CD4 receptors. MHC class 1 interacts with CD4 receptors such that whenever, uh, you know, let me just draw the structure. These interact with CD4 receptors. MHC class 1 interacts with the cytotoxic T cells as well as natural killer cells, whereas cytotoxic T cells and macrophages, whereas MHC class 2 interacts with CD4 receptors, as a result of which it is found in antigen presenting cells. Likewise, if we talk about other class, if we talk about other structure, let's suppose we are having now coming to the point that here when MSC class 1 of natural killer cell had the antigen represented over it, that antigen represented over it will be the sent towards either to the B cell, or it will be sent towards the T helper cell, where the T helper cell will further receive it through its receptor, which is known as the T cell receptor. That T cell receptor, when would be receiving it, it will cause further, uh, you know this T cell receptor will cause further to release interleukin 2. It will cause to release interleukin 2. Of course, there's a separate mechanism for it as well, that here interleukin 2 will be released out of the T helper cells, as a result of which the B lymphos, as a result of which now here as macrophages will now natural killer cells are present. This work can also be done by macrophages, but when macrophages will be performing this function, they will release uh, here and natural killer cells, in order to stimulate the helper T cells, will release interferon gamma, but for macrophages will release interleukin 1 uh, in order to stimulate the helper T cells. Now, when helper T cells release interleukin 2, they stimulate the cytotoxic T cells as well as the B cells to come and take the antigen from the helper T cells. So, such that helper T cells now represent that antigen over its MHC class 2. It represents that antigen or MSC class 2, and when it represents that antigen on MSC class 2, that antigen, when representing on MSC class 2, will now be presented over the B cell receptor of the B cell and T cell receptor of the cytotoxic T cell. Once they gain the antigen, present the antigen over B cell receptor of B cell and T cell receptor of cytotoxic T cell, it will cause the cytotoxic T cell to generate a cell mediated immune response against it, and then B cell to generate humoral immune response against it. Now, what does it mean by the cytotoxic T cell response? Cytotoxic T cell generates a response by basically causing the cytotoxic T cell generates a response. Cytotoxic T cells generate a response by directly attacking the particular type of particular by directly attacking the particular type of pathogenic organism having that antigen, and at that moment it will release perforin protein as well as gay enzyme in order to destroy it. And then some T memory cells will also keep the record of that encounter. Moreover, when it comes to B memory cells, after receiving the antigen presented over there B cell receptor, they will tend to differentiate and proliferate. They will proliferate and differentiate into B memory cells and B plasma cells. B plasma clone cells will cause to secrete antibodies against that particular type of pathogen containing the antigen, as well as the B memory cell will keep a record of that antigen, so that in future whenever the infection is again causing to attack our body, then that pathogen can be easily destroyed. And whenever there's a future encounter, the B memory cells as well as the T memory cells, the B memory cells, whenever detect that there's a future encounter, the B memory cells will directly convert themselves into uh, plasma clone cells, due to which antibodies will be secreted from plasma clone cells. And when T memory cells will see that if there is a pathogen again coming in the body, then T memory cells will now convert themselves into the pathogen and convert them into the cytotoxic T cell, due to which they will destroy the the pathogen which is causing the infection. Now, this is how it works. One more thing that I would like to highlight over here, this is on the basis of the MSC class 2, because MSC class 2 is associated with CD4 receptors and its co-receptors, as well as the MSC class 1 is associated with helper T cells and macrophages. It is associated with no, actually MSC class 1 is associated with cytotoxic T cells, and MSC class 1 associated with cytotoxic T cells and natural killer cells. These are associated with natural killer cells, not with uh, helper T cells and macrophages, because helper T cells and macrophages are CD4 cells. They are basically associated with MSC class 2. MSC class 1 is associated with cytotoxic T cells and NK cells. So this is all about the introduction. Now coming to a basic introduction of some T and B lymphocytes. T and B lymphocytes here first and foremost includes a blood stem cell. We have a blood stem cell which is known as erythrocytoblast pluripotent stem cell. That pluripotent stem cells generally divides into five major classes of cells, which is known as the monoblast, lymphoblast, megakaryoblast, proerythroblast, as well as myeloblast. So uh, we can also call them myeloid stem cell, lymphoid stem cell. Depends on how we utilize the terminology. Now, when it comes to uh, lymphoblast, lymphoblast basically further divide themselves into you know lymphoblasts divide themselves into prolymphocytes. Prolymphocytes mature themselves into uh, prolymphocytes are differentiated into larger lymphocytes. Then larger lymphocytes either go into the bone marrow for maturation to convert into convert into B lymphocytes, or they go into the thymus for maturation to convert into T lymphocytes. Some of the maturation also occurs in spleen and lymph nodes, but majorly that occurs in thymus. And majorly it occurs in thymus, but some occur in lymph node and spleen as well. Now, this is a basic introduction. A few MCQs point I would like to highlight, that here in the blood there are around 4 to 11 into 10 s power 9. 4, let me just write that down. 4 to 11 into 10 s power 9 into 10 s power 9, 4 to 11 into 10 s power 9 per liter of leukocytes. If it is below 4 into 10 s power 9 per liter leukocytes, then that condition is leukopenia. If it is above 11 into 10 power 9 per liter leukocytes, that would be leukocytosis. Likewise, if we are having um, the concentration of platelets, that would be around 250 to 500 into 10 s power 9 platelets per liter. If it is below this range, then that would be considered as a condition of polythrombocytopenia. Thrombocytopenia. One more thing, if we are having concentration of RBCs, concentration of RBCs in the blood includes 4.2 to 6.4 into 10 s power 12 per liter. In male, generally in male the average is 5.5 into 10 s power 12 per liter. In female it is around 4.8 into 10 s power 12 per liter. And when it comes to if it is below the range of 4 into 10 s power below 4 into 10 is power 12 per liter, then this condition is known as anemia. If it is above 4, if it is above 6.5 into 10 is power 12 per liter, that condition is known as polycythemia. So this is a basic intro about it as well. Now this is just a diagram in order to make you understand a few of the concepts. Now here, cytotoxicities, there are three major, there the these lymphocytes are divided further into their types. T lymphocytes, when mature in thymus, further divide into their types, which are known as T helper cells, T killer cells, uh, T killer cells are also known as effector cells, and T memory cells, also known as you know, T memory cells are present there as well as suppressor T cells, also known as regulatory cells. And here, when we come to talk about these cells, um, we have generally divided them into two major categories: CD4 cells and CD8 cells. CD4 and CD8 cells. CD4 cells basically are those which have a CD4 receptor as well as co-receptor C into CR4, such as T helper cell is a CD4 cell, as well as uh, a T helper cell is an example of CD4 cell, whereas a CD8 cell includes example of cytotoxic T cells as well as example of cytotoxic T cell. It is having CD8 receptor, and suppressor T cell or regulatory cell, which is having example of CD8 cell. After that, uh, when it comes to the function of each cell, helper T cell, as previously told, these release interleukin 2, which causes to, you know, help interleukin 2, they causes to release interleukin 2, which causes to stimulate the cell mediated immune response by stimulating the cytotoxic T cells, and humoral immune response by stimulating the humoral cells, or which are known as the B lymphocytes, as a result of which they play an important part in generating immune response. After that, we are having cytotoxic T cells. These are CD8 cells, contain CD8 receptor. One more thing, the helper T cells are having co-receptor C into CR4. And when it comes to cytotoxic T cells uh, basically directly attack the cell by releasing proenzyme, uh, perforin and grain enzyme, sorry. And then they also release tumor necrosis factor alpha, as well as interferon gamma in order to stimulate other cells, such as in order to stimulate other cells such as macrophages and dendritic cells, as well macrophages and dendritic cells, to come and attack over the cells and generate effective immune response. Moreover, when it comes to suppressor T cells are those cells which inhibit the activity of helper T cells, as well as the cytotoxic T cells, as well as the B plasma cells, so that they may not destroy our own body cells. After that, when it comes to the last category, which is a T memory cells. T memory cells are those cells which keep a record of the attack which has been made by the cytotoxic T cells over the particular type of pathogen against it. So this is how we do it. This is the classification of T lymphocytes. But when it comes to B lymphocytes here are when they get the antigen presented over their B cell receptor. One more thing I would like to highlight over here, that here suppressor T cells. Suppressor T cells are also having CD8, CD, and TCR receptors. These are having three receptors, and natural killer cells are having CD56 receptors. Anyways, B cell receptors are present on B cells, such that when the antigen gets represented over the B cell receptor, they differentiate into B plasma clone cells, as I previously told, which secrete antibodies, as well as the B memory cells. Now, B plasma clone cells are those which include secrete antibodies such as IgA, There are five classes of antibodies: IgA, IgG, IgD, IgM, and IgE. So uh, we are going to discuss that in our next part of the video. So here, B plasma cells create antibodies, whereas B memory cells create a record of that antibody, so that in future, if there's any infection, the B memory cell will cause to convert themselves into B plasma clone cell and themselves produce the antibodies. So this is the function of uh, B cells, as well as one more thing, that here, as B cells are important, these are B cells are named after they are named because they were found in the cloaca of chicken. There was a structure in the cloaca of chicken, a glandular sac-like structure known as bursa fabricus, from where the scientists discovered it, and then it was named as B cell. And but in humans, it is matured in bone marrow, right? And apart from that, this is a general intro of it, and yeah, this is all about this video. Hopefully, we'll meet again in another one.